go ahead and go on to our last speaker, Akshay. How do I pronounce your last name? So I do this correctly.
Yes, so it is Akshay Binayke.
Okay, excellent. Well, Akshay is an early career immunologist who is currently pursuing his PhD under the supervision of Professor Amit Alshwashi at the Translational Health Science and Technology Institute in India. He specializes in T-cell immunology and inflammatory diseases. And during the COVID-19 pandemic, he responded to the national call and gained expertise in antigen-specific T-cell assays, helping to test the cellular immunogenicity of different vaccine candidates in phase 2 and 3 clinical trials. His team was one of the first to demonstrate that the T-cell immune response was largely preserved against the Delta variant, even though the antibody neutralization was ineffective, thereby providing the basis for reduced severity of COVID-19 infected individuals during the Delta surge. As part of his PhD thesis, Akshay is investigating the molecular mechanisms that govern the generation and function of T cells in inflammatory diseases, and has identified several small molecules that ameliorate psoriasis in mice models. For his work on COVID-19 immunology, Akshay has received the G.P. Taller Young Scientist Award, the highest award for early career immunology researchers in India. His title today is Proinflammatory Innate Cytokines and Distinct Metabolic Signature Shapes the T-cell Responses in Active COVID-19.
Thank you, Emily. Thank you for such a wonderful introduction. I hope my slides are visible to you all. Okay, so I belong to this institute of THSTI, which is located in New Delhi. And as mentioned, the title of my presentation is Pro-Inflammatory Innate Cytokines and Distinct Metabolic Signature Shape the T-cell Response in Active COVID-19. So we all know how devastating effect the COVID-19 had in all our lives. Almost 6.8 million people have succumbed to this disease and still counting. So understanding this disease is extremely important, and finding therapeutic avenues is of critical importance. So in the early stages of the pandemic, we conducted a test negative case control study to understand the real world effectiveness of ChAdOx and COVID-19 against the Delta variant. And we found out that full vaccination prevented moderate to severe COVID-19 in 81.5% of the individuals, despite significant reduction in the neutralizing antibody responses against the Delta variant. So we tested T-cell responses in 60 number of individuals and performed T-cell studies. And we found out that the antigen-specific T-cell responses were largely preserved among the ancestral and the Delta variant, providing the basis for reduced severity for COVID-19 vaccinated individuals during the Delta surge. So another important study was published by the Bertoletti et al., showing that the individuals who have had a mild disease tend to have many functional T cells at the early stages of infection, whereas those individuals who have a severe disease tend to have very few functional T cells. And no such correlation was observed with the antibody kinetics in this cohort, therefore suggesting that T cell responses are particularly critical for protection against the variants of SARS-CoV-2 as well as against severe COVID-19. So our conventional virological immunologic wisdom suggests that mild infection is resultant when a strong and timely innate immunity takes control and results in robust T cell response, and the viral load is contained in time, resulting in mild infection. Whereas in the case of severe infection, there's a poor innate immune response, resulting in poor T cell response, and the viral load is not contained within time, resulting in severe infection. Therefore, keeping this thing in mind, we wanted to understand the molecular cues, the innate and metabolomic signatures that contribute to a robust T cell response. And to do so, we followed a cohort of mild COVID-19 patients and followed them up at 3 different time points longitudinally and collected blood samples. The time points were V1, which is day 0 to 3 of positive PCR report, V2, that is 1 week, that is around 7 to 9 days after positive PCR report, and V3 is 14 to 16 days after positive PCR report. So we collected a blood sample at each time point, isolated the plasma and performed untargeted plasma metabolic analysis, And we also collected the PBMCs. Now, these PBMCs were used for two different things. One was to identify the innate immune responses at the early stages of infection by stimulating them with TLR378 agonists. And we also studied the spike-specific T cell responses by stimulating the PBMCs with spike peptide. And we tried to correlate all things with another, such as the innate immune responses were correlated with the T cell responses at V3, and the metabolomic signatures were correlated with the immune responses of the innate and adaptive at V1 and V3. So as obvious as it can be that as the time went ahead, by V3, the antigen-specific T cell responses increased significantly. This can be observed from the expression of CD4 positive IFN gamma positive response and the CD8 IFN gamma positive responses, which was significantly increased by V3. And so was the case of activation-induced marker responses, which are the CD137 OX40 expression on the CD4 cells and CD137 and 669 co-expression on CD8 T cells. We saw that particularly the CD4 responses increased after significantly compared to V1, that is compared to day 0 to that of days 14 to 16, the CD4 T cell responses were significantly increased. So was the scenario in the CD8 responses, but the magnitude of response was slightly reduced in the CD8 compartment. We also studied the cytokine releases in the form of particularly the type 1 specific cytokines such as the IFN gamma, TNF alpha, IL-2, which were elevated and increased by V3. So was the cytotoxic cytokine granzyme B, which was significantly increased by V3. We also tested if these T cell responses generated in the active COVID-19 individuals stimulated or exposed to the first wave of COVID-19, that is the ancestral strain, were able to cross-react with those of the Omicron and Delta spike. This is a unique scenario where the active COVID-19 individuals were tested for their cross-reactivity. And we saw that the cross-reactivity with the Omicron and Delta was largely preserved, but to some extent reduced, as we can see in the CD4 compartment and also in the CD8 compartment. The next approach was to identify the innate immune responses. And to do so, we took the PBMCs from V1 and stimulated them with the TLR378 agonists. So the TLR78 agonist, which is R848, mimics the presence of single-stranded RNA. And the TLR3 agonist, which is poly-IC, which mimics the double-stranded RNA, kind of mimics in vitro the presence of virus, and this results in stimulation of the antigen-presenting cells and the innate immune cells for 24 hours, and the culture supernatant was picked up and cytokine analysis was performed. So what we could observe was that when we performed correlation analysis of the T cell immune responses at V3 with that of the TLR-specific innate immune responses at V1 and the humoral immune responses at V3, that the cytokines, which are the IL-1 beta, IL-23, and IL-18, were strongly correlating with that of the antigen-specific T cell responses at V3. When we looked further in closer, no such correlation was observed in the humoral immune responses. Neither was any response or correlation observed with the other cytokines such as IL-5, IL-10, and IL-8. So we looked closer in detail about these pro-inflammatory cytokines, and we found out that these cytokines were particularly and significantly getting correlated with the type 1 specific immune responses, such as the IFN gamma release of CD4, CD8, the IL-2 release by CD4 cells, the release of granzyme B was also correlating significantly and positively with that of IL-23, IL-1 beta, and IL-18. And so was the TCR-specific stimulation responses, such as the CD4 AIM and CD8 AIM responses. Therefore, what we could conclude was that during mild infection, a strong and timely innate immune response upon TLR stimulation in the form of a pro-inflammatory cytokine response of IL-1 beta, IL-23, and IL-18 gives rise to a robust antigen-specific T cell response in mild COVID-19 infected individuals. Next, we wanted to understand what is the metabolomic signatures in COVID-19 patients. And we found that the COVID-19 patients, which are represented by this PCA plot in green and blue region, were quite distinct from the metabolic profile of that of the healthy individuals. You can see in this heat map how this region, which is represented by the healthy individuals, is quite distinct from that of the COVID-19 acutely infected individuals. And therefore, this led to look further and identify some metabolites which might be correlating with that of these pro-inflammatory innate immune responses at V1 and the antigen-specific T cell responses at V3. And what we could find out was that some metabolites which are particularly involved in the arginine biosynthesis and D-glutamate and glutamine metabolism at the V1 stage were correlating significantly with the pro-inflammatory innate cytokines such as the IL-18, as well as with the antigen-specific T cell responses, such as the cytokine response upon peptide post-stimulation in the form of IFN gamma release or the TNF alpha release. So was the case with V3. But what we could do was we performed a pathway analysis of all the plasma metabolites, which were significantly correlating with immune responses, which was innate as well as adaptive. And when we did this pathway analysis, we identified that all the metabolites involved in arginine biosynthesis were enriched in these significantly correlating metabolites. So was the case when we were studying the plasma metabolic signature of the V3, that is the later stages of infection. The metabolites similar to those with the that of the V1, the metabolites involved in arginine biosynthesis, such as the L-citrulline, itaconate, were correlating significantly with the antigen-specific T cell responses, such as the CD4 IFN gamma, or it was also in the case of CD4 AIM, the IFN gamma release, and also the pro-inflammatory cytokines, such as the IL-23 and IL-18. And when we did this pathway analysis, again, the arginine biosynthesis metabolomic pathways were getting enriched in the plasma metabolites of those individuals who could generate a robust T cell response and also give rise to a robust and strong pro-inflammatory cytokine response upon TLR specific stimulation. So what we could conclude was that a early innate immune response in the form of IL-18, IL-23, and IL-1 beta, along with a plasma metabolomic signature involving arginine biosynthesis, could give rise to a strong antigen-specific CD4 and CD8 T cell response during mild COVID-19 infection. And this particular scenario can be replicated in novel vaccine platforms where TLR agonists, which particularly stimulate or give rise to a pro-inflammatory cytokine response in the form of IL-18, IL-23, and IL-1 beta, may result, along with a metabolomic supplementation enriched in metabolites involved in arginine biosynthesis, may result in a better protective T cell response against COVID-19 and its variants. Now, this particular cytokine signature of IL-23 and IL-1 beta might be quite intriguing, that these are the pro-inflammatory cytokines, and how can they give rise to a good Th1 specific response? Because these might skip the tend the balance towards a Th17 specific response. But a recent paper by the Vijay Kuchru group quite made it clear that cytokines such as the IL-23 and IL-1 beta, particularly IL-23 receptor, gives rise to a Th1-like biased approach and a Th1-like phenotype in the T cells. And this particular observation in our cohort kind of supports this particular scenario where a strong antigen-specific Th1 biased CD4 and CD8 T cell response might be a scenario of observation. So I'd like to acknowledge, most importantly, my supervisor, Dr. Amit Awasthi, and other co-authors of the study, such as Dr. Shailendra Mani, Dr. Yashwant Kumar, Dr. Tripti, Ayman Jyotsna, Manas Upasna, and my entire lab, and most importantly, my institute and the funding agencies who made this study possible. And I would also like to thank the volunteers who have provided blood for this study, and the Professor A.K. Pandey from ESIC Hospital, who made this provision of providing blood samples from the patient cohorts quite possible. So thank you all for listening and your attention. I'm open for any questions. Thank you.
Thank you for that, Akshay. Very nice presentation. Are there any questions? Yes, Jeremy, go ahead.
Akshay, very much for your talk. That was great. One of the things I was just sort of interested in at the end there was the cytokine upregulation, that you were discussing. Obviously, those particular cytokines, just some of many that could be activated in the context of innate immunity, and other TLR agonists, presumably also in general terms, obviously going to upregulate innate immune activation to then lead on to appropriate and robust T cell responses. How important do you think it is that these IL-1 beta, IL-18, IL-23 are the kind of the key ones? Because presumably, is this more like a virus-specific situation, or do you think that ultimately, as the innate immune system is, it's kind of going to need to be capable of responding to all types of onslaughts, immunological onslaughts? So it kind of needs to be to ensure a robust T cell response, regardless of the type of virus. I'm just wondering what your thoughts were on that.
Yeah, that's a very, very important question and very nicely put up. So the cytokines, particularly in this scenario, what we could find was these particular cytokines were correlating. It was quite intriguing that the type 1 interferons were another important aspect, which has been considered quite important in the case of COVID-19 immunity. But there was one paper which was published in Cell Reports Medicine, which suggested that the type 1 interferons did not correlate with the antigen-specific T cell responses. In this particular study, we wanted to look into the factors or the cytokines which are particularly important in Th1 specific biased specific responses, and which were protective in nature in the case of COVID-19 infection. In the case of maybe some other infections which require a Th2 biased responses or a Th17 biased responses, some other cytokines might be important. But probably from what we could understand from our analysis, what we could see was that these cytokines were specifically important in the case of, and these cytokines particularly upon TLR stimulation and at the early stages of infection. These are quite important to be released at the right time and not after a prolonged exposure to the virus or something like that.
Great, thank you.
There was another question, but maybe not. Okay, well, great, thank you. If there are no other questions,